Welcome to the Advanced Concepts presentation number 4 by myself, Mariella Roper. This presentation will discuss the steps involved in generating acetyl CoA from pyruvate by explaining the structures and enzymes of each step in this complex. The generation of acetyl CoA from pyruvate is a crucial step between glycolysis and the beginning of the citric acid cycle. Pyruvate is produced from glucose in glycolysis and marks the beginning of aerobic respiration. In the absence of oxygen, pyruvate is converted into lactic acid, but in the presence of oxygen, pyruvate is converted into the molecule acetyl coenzyme A, or acetyl CoA for short. It is then acetyl CoA that enters the beginning of the citric acid cycle. Thus, the generation of acetyl CoA from pyruvate marks the beginning of aerobic respiration. Because the formation of acetyl CoA is the beginning of aerobic respiration, it cannot take place in the cytoplasm of the cell like glycolysis. Instead, pyruvate must be transported to the mitochondrial matrix where it can begin to undergo aerobic respiration. In the mitochondria, pyruvate is oxidatively decarboxylated by what is called the pyruvate dehydrogenase complex in order to form acetyl CoA. The conversion of pyruvate into acetyl CoA consists of 3 steps: decarboxylation, oxidation, and then the transfer of the resultant acetyl group to coenzyme A. This process requires the sequential action from 3 enzymes and 5 different coenzymes, known as the pyruvate dehydrogenase complex as mentioned before, and the steps must be coupled in order to preserve the free energy derived from the first step in the process. The first steps of the process, decarboxylation, is the rate-limiting step in the reaction and is catalyzed by the pyruvate dehydrogenase component, or E1 for short. This requires the help of coenzyme TPP, or thiamine pyrophosphate. In this first step, pyruvate combines with the ionized form of TPP and is decarboxylated to yield hydroxyethyl TPP along with CO2, since decarboxylation is the removal of CO2 from a molecule. The second step of the process is oxidation, more specifically the oxidation of the hydroxyl or hydroxyethyl group attached to TPP from the previous step. Also, like the previous step, this reaction is catalyzed from the same enzyme, so is also part of the E1 complex. The oxidation forms an acetyl group, which is simultaneously transferred to the second cofactor, lipoamide. The disulfide group of lipoamide is reduced, and the entire second reaction yields acetyl-lipoamide molecules. The third step of the process is the transfer of the acetyl group to coenzyme A. This process involves the second enzyme in the pyruvate dehydrogenase complex, namely the dihydrolipyltransferase, or more simply E2. E2 catalyzes the reaction as the acetyl group from acetyl-lipoamide is transferred to CoA to form acetyl CoA. Additionally, the thioester bond formed in the previous step is preserved, and acetyl CoA is now ready to be used as fuel for the citric acid cycle. Although the generation of acetyl CoA from pyruvate is now complete, the process must be reset in order for another pyruvate molecule to bind and generate acetyl CoA again. Thus, the fourth step takes place and is catalyzed by dihydrolipyl dehydrogenase, also known as E3. In this step, dihydrolipoamide that was generated alongside acetyl CoA must be oxidized back to lipoamide. This step also makes use of 2 cofactors, FAD and NAD+. Dihydrolipoamide transfers 2 protons to FAD, which then in turn transfers a proton to NAD+ to form NADH. Then, dihydrolipoamide is successfully oxidized back to lipoamide and gives the ability for another pyruvate molecule to bind and start the process over again. As an incredibly important process, generating acetyl CoA from pyruvate is the irreversible reaction that bridges glycolysis and the citric acid cycle. The entire process makes use of 3 enzymes and 5 cofactors, with the first 2 steps making use of E1 alone. The entire process of generating acetyl-CoA is the crucial beginning in aerobic respiration, allowing organisms to generate as much energy as possible from each molecule of glucose.